Welcome to the Maine Science Podcast. I'm Kate Dickerson. Today's episode is a conversation I had with Jason Bubier, Senior Research Scientist at the Jackson Laboratory. I was absolutely mesmerized by Jason's 5-Minute Genius talk at the Maine Science Festival this past March, and I knew that I had to talk to him about his path in science as well as his research. His study of the microbiome and how it can impact genetics is fascinating. It's also one of the best examples I can think of that shows how much we still don't know about the world around us and how vitally important basic science research is to help us figure it out. Our conversation was recorded in May 2026. I hope you enjoy it as much as I did. Jason, welcome to the Maine Science Podcast. I am over the moon to have you. It was a lot of fun to listen to you talk at this past year's 5-Minute Genius. And that, as it often does, inspired me to want to talk to you more about your background and how you got into science. You're doing really extraordinary work at the Jackson Laboratory. But before I let you talk about that, what I'd really like to know is, how did you get into science? You did mention actually either at the event or later that you were a first-generation college student, which makes your journey to Jax all the more interesting.
Yeah, thank you, Kate, for the opportunity to come on here and talk with you about my journey to get here. And it's very exciting for me to be able to share, kind of feeling like a success story of having come from one place and ending up in another. So I grew up in a world of working class. Father was a truck driver. Mother was a stay-at-home housewife. They are from Maine. We still have our 1822 farmhouse in western Maine and our own little family cemetery as well. So we're definitely rooted, but they moved to Connecticut in the 60s looking for jobs. A lot of the mills in that area, the woolen mills and the match factory and stuff, were all closing down. So my father, also not really wanting to work in the woods, which is what his father did a lot of, was a lot of logging, and he realized it was a lot of work. He was trying to find something different, so we moved to Connecticut. And thankfully, I got to benefit from them settling in a town with a wonderful school district. And this is one of the things that's, I think, most important to me, is the fact that education is a way to move within classes, if there are even classes or social structures within the American system. The school district that I went to is Regional District 13 in Middlefield, Durham, Connecticut. The school had school choice, but it was choice in that you had a choice between two different programs. You could have a traditional classroom program in which there's desks and you change classes, starting in first grade, you have a science class, a math class, etc. Or you could have this integrated day type program, an alternative classroom, it was called, an integrated day classroom. It was also a higher order thinking skills classroom. And so the classrooms were set up very differently. Instead of desks and chalkboards, there were learning nooks and couches and beanbag chairs in the classroom. And a lot of the learning was student directed. And so you got to really follow the things you were interested in. And I know that like in 5th or 6th grade, I really got interested in science and was set up and did a project on growing peas. I grew peas, and my hypothesis was if I grew them in the light, that they would grow better than if I grew them in the dark, because plants need water, soil, and sun to grow. And every day I'd come in and I'd measure my peas. And the interesting thing here, and this is science, is that the ones in the dark grew taller and faster because of what I learned years later is a process called etiolation. Etiolated, E-T-I-O-L-A-T-E-D, etiolated, which means that the hypocotyl, the distance between the base of the stem and the first leaf, really extends as the plant is trying to find the light. So in the absence of light, the plant actually grew a longer hypocotyl and was actually taller in the dark compared to the light. So this just blew my mind. It turns out like maybe the output from my study shouldn't have been the length of the stem. It should have been maybe how long it takes to get to a flower or something, but it was a short-term project, and that was a nice output to measure, was the hypocotyl length. And in this particular arts integrated classroom, like we made our own little project books and presented all this data. We made graphs that would represent all of the data that I collected in my project, and then write text about it in these little project books that we would make. I found science and math came easily to me because a lot of it was visual, like lots of graphs and things like that. And it turns out in 3rd or 4th grade, through the standardized testing, it was determined that I had a learning disability, that I was actually behind in reading. And so we activated the early intervention programs that I had at the time and caught me right back up on my reading within 4th, 5th, 6th grade. I got up to grade level. But it makes sense to me that I really did hone in on those visual things. When the letters weren't making as much sense to me, the numbers and the charts definitely were clear, and I knew what was going on.
I'm amazed that the integrated classroom was an option from the get-go. Was that something that your parents knew that would appeal to you? I'm assuming first grade Jason didn't have the whole choice in what was going on.
Oh, yeah, I had no idea. One of the things is that they were smaller class sizes. The first and second grade were combined together. So my mom knew that I didn't really like change. And so having the same teacher for 2 years could be a benefit to me. It was a lot of hands-on learning and a lot of doing. And she was very much into doing and hands-on too. And so she kind of was following her own learning style and was like, this would benefit me. Maybe it'll benefit him. This is in the 80s when computers are just coming online in the Apple II series of computers. And I was like saving Campbell soup labels to get computers for my classroom. And one of the things that happened is I became really good at using computers in the basic and the logo programming languages of the time. And I was offered the opportunity to help teach another kid about computers. And he was in like a second or third grade. He was a year or two behind me. And he was the superintendent's son. And that just made quite an impression on me because my parents respected the superintendent, respected the school district. And then here I was, my dad, a truck driver, and the superintendent's son, and I'm teaching him about computers. It was just like really eye-opening and a great experience. And it kind of made me aware of like, you can go where you want to go, you can do what you want to do. And if you're into something or passionate, people will want to learn about it from you. And it turns out that that kind of followed me through high school. I was really into science in biology and AP biology, into the plants. Again, we had the rapid cycling Brassica rapa, or the fast plants, that you could cross a dwarf with a tall and see what happens and do your Punnett squares. And so I was really into that. But at the same time, I was also into the library. Media science was my issue because I was very into the technology in the library that was coming online and the ability to send messages from one computer to another over a local area network. It was crazy. I was just so into it. And so that's when I actually moved into PCs, not Apples at this time, and DOS, or the disk operating system terminal, and that type of coding.
We're going to have to make sure listeners younger than 30 do some looking up of what we're talking about here because it's going to be foreign to them.
Yeah, this is in the beginning of technology evolution, like pre-internet, starting local nets and then going out to larger area networks.
So I'm curious, I saw that you went to UMaine Farmington. Was that because of the family history of being in Western Maine?
That's right. My family retired to Maine right when I graduated high school. My father was a union job, and so he put in 30 years and was able to finish and check out and retire back to his family homestead. And I got in-state tuition by being in Maine, and it was close by my parents' house.
That must have made the transition to being the first in your family to go to college a little bit easier. So my husband is a professor at UMaine, and one of the things that, from an outside perspective, that I think UMaine does really well is they're aware that there's a lot of first-generation college students. And that's not easy to be the first to do a path when your primary support structure doesn't know what that path looks like.
Yeah, I agree totally. Going on campus, I was learning about things like associate's degrees, bachelor's degrees, PhDs, masters. What are these? Going to the bursar's office, and just the terminology of campus life was just unfamiliar to me. And my parents had no idea to prep me for this. So I just walked onto campus as a first generation student and was so excited to see all the science classes that I was going to be able to take. And UMF was a really great fit for me because it was small. They didn't have a lot of hands-on research, which I was really concerned about because I learned quickly that to get anywhere in science, you needed to have a good lab experience before you're going to get into graduate school. And then I learned what graduate school was and that the possibility that I could go there and they would actually pay me to go to school through an assistantship. My tuition would be waived and I'd get money. And that blew my mind and blew my parents' mind. Like, more college and you don't have to pay for it. So they thought it was a great setup and were very proud of me. I remember my graduate school acceptance letter being on the mantle for a while of the family fireplace because they were just so proud that I got in.
When did you realize you wanted to keep going past a bachelor's degree?
Pretty much as soon as I figured out that when you went to college, the college was more than just one thing, that there were advanced degrees and that there were these professors that I was hanging out with, making salsa on Sunday with at club events, and they had PhDs and what a PhD was and how they could like dedicate their life to science. In high school, I did an internship with my aunt. She was a lab tech at a hospital. So we had lots of stool samples and other urinalysis to do. And so I spent a shift with her, and that was enough of doing some routine lab tech stuff. I loved all the lab stuff, But it was all routine. Nothing was really discovery based, creating new things. They just came in and followed the same protocols every day. And that didn't seem interesting enough to me. I was all interested in how did they get to this protocol? Where did this come from? Why are they doing it this way? And is there a better way? So that was my high school internship experience. Biology is definitely where I want to go. And then when I got to college, I saw like, okay, so there are other degrees, PhDs, and they can do research and you can get paid, you can write for grants, and the government will pay for you to explore things that you're interested in. And it just seemed like a great thing to discover.
Yeah, it's so interesting. Your story about the pea plants in fourth grade, you were already clearly wanting to figure out how things worked as opposed to just using programs that people have already figured out. And that's great, right? Like you figure that out one way or the other. Sometimes you just want to do what people have already figured out and do it really well. But to know there's another path is pretty great. All right, I'm going to jump ahead to your work at Jax because it is just wildly fascinating to me. But to get us there, you were doing a lot with genetics, and that is such an all-encompassing category. I think it requires, in this case, kind of an explanation of how you're using it. I mean, you are doing basic research, but you're doing it in a way that's very different, I think, than most people think about with genetics. And then before you answer that, you were in that sweet spot of someone who I'm thinking you were in the midst of your PhD when the genetics revolution really happened, right? Like the moonshot. You are living in this truly golden age of biology and genetics research.
Yeah, it was great. The Human Genome Project was happening. We were getting the sequence of it. When I got into graduate school, I also got an internship through the Mills family to do some research in a technical lab at the Jackson Laboratory back in the 1990s. And so I went there, and one of the things I did was extract DNA, and then I was trying to clone it into different vectors. And my sister-in-law was like, really? That shouldn't be too hard. They find it all the time around a crime scene. And so it was like, yeah, there's disconnect between what the public knows about science and what is actually happening in the lab, and that getting this purified DNA to be able to digest and do molecular biology on was different than just, kind of forensics evidence, CSI stuff. The question is about genetics and how I'm using them. So I think that we're looking at it at the population level in our lab. So one of the things that we're looking at is within any population, there's a lot of variation across different subgroups. And in mice, there's different inbred strains that we look at. Each strain that we look at can be inbred, and then that means that the genetics are going to run true, kind of like breeds of dogs. So we have breeds of mice, kind of like you have breeds of dogs. And some breeds of mice are going to be very sensitive to certain compounds or drugs, and others are going to be more resistant to them just naturally. And so we can control the mating, unlike in the human population, and we can control who is mating with who. So a high consumer of alcohol can be mated to a teetotaler, and we can see how the trait carries out in the progeny and seeing which mice are carrying the trait of being interested in drinking a lot versus the trait of not being interested in drinking a lot. And one of my big ideas here in the molecular biology of addiction is to look at how is the environment, the genes environment regulated together, because it's never nature or nurture, it's nature and nurture, genes and environment. And at Jackson Lab, we can keep the environment very stable, but one of the things that can vary is the microbiome, or the microbes that live in the guts of the mice. And there's a genetic component to that, and then there's also a diet component, but all our mice eat the same diets. So we'll have different mice with different genetics and different microbiomes. And I'm understanding how does early life events or other exposures in their past affect their microbiome, and how does that affect their behavior? If they received alcohol in utero during gestation, like a fetal alcohol syndrome model, how does that affect the microbiome? How does that affect their development? And so we use this population, and it really does resemble a human population in that it captures the large diversity of SNPs or nucleotides that are varying across what they call the pangenome. When they sequenced the genome, they just sequenced one guy's genome. And that was our reference, but that one white guy doesn't represent all of humanity. So we got to create the pangenome where you look at all of the genetic variation. And that's what we do with this mouse population is we look at all of mousedom. And we do experiments to see, do we do a finding in one strain? Does it translate to another strain? Before we try to cure a human disease, can we cure it in another strain of mice? Will those mice that are very sensitive to the drug, are they going to be sensitive to treatment too? Or are they going to be resistant? What's going to happen compared to a John Doe mouse that we have is like our workhorse called Black 6, C57 Black 6J. That's a standard kind of inbred strain that's used for a lot of research. A lot of our discoveries in neuroscience around the world have been made in that one strain, but how well do they relate to other mice? And then eventually, how do they relate to humans?
So the microbiome thing is honestly wild to me that it can vary so much from mouse to mouse or person to person. And I'm curious, first, maybe describe what a microbiome is just to make sure that everybody knows what we're talking about. But also, has that always been known or was that figured out? Like, okay, so let me give you a visual. I imagine two mice in the same cage. I don't know if that's actually how you do it or not. And completely identical. They're essentially clones. Their microbiomes could actually be pretty different, right?
Uh, there could be some specific differences, but their microbiomes are pretty much going to be similar because they're cage mates, and mice engage in coprophagia where they're eating each other's feces. So they are constantly co-inoculating each other with each other's microbes.
Actually, that's really cool. So if they were the same species, like the same kind of clone, but in separate places, then they would have very different microbiome.
Then you can start to see some drift and some specialization, usually based on diet or how much of the bedding are they eating, because the bedding can actually become a source of fiber for fermentation for the different microbes. And so it depends on kind of their behaviors as to what their microbiomes are going to look like. But yes, two mice housed together definitely will have similar microbiota. But you could have genetically different strains in the same cage, and then they would be close to each other, but there would be some very specific microbes that one mouse could harbor and another mouse wouldn't harbor. So there are genes basically for resistance to colonization. You could be resistant to being able to be colonized by Staph aureus or something. Or you could be sensitive to that based on your genome. So your genome can encode what's in your microbiome. And by microbiome, I'm talking specifically about the microbes, the bacteria, and the archaebacteria that are found in the gut, the gastrointestinal tract, which is an ecosystem from one end to the other. There's a variety of different levels of oxygen. So there's a variety of different aerobes and anaerobes as you go down the gut. And the microbes that I'm interested in are mostly, as I said, the bacteria and the archaea. There also includes the fungi and the viruses that are also part of your microbiome. But this is something that is set up at birth during a traditional vaginal birth. This is often when the pups or the baby receives an inoculation of the mother's microbiome. And that's when it starts the colonization. And that starts with the similarity that you'll see as a transfer from the mother to the pups.
So when was that figured out that the microbiome was that vastly different and then so tied to genetics?
Yeah, I can't really put a date on it. I think that because it's so complicated and varies so much, a lot of people, a lot of researchers in the field don't want to embrace it as much as they want because it adds a lot of noise to our data and our analysis and is a potential source of variation that they're not accounting for. So a lot of people are not really focused on the microbiome, but there is variation that's occurring there. And we've been in our populations been able to identify loci that when you have one locus, you're going to be able to be carrying this bug. And then if you have a different version at that same locus, you won't be able to carry that bug. So that's the heritability, and it's been shown in human studies for a long time.
So this does make research intensely complicated. What is it about this that has appealed to you to make your life filled with this kind of noise that you're trying to decipher?
Part of it is the interest between genetically identical mice at the Jackson lab will not always behave genetically identically. Like only 50% of that strain of the black 6 mice are we able to get addicted to cocaine, for example. Another half, we can't get addicted. They won't reach the criteria of self-administering the cocaine to themselves. So what is the cause of this variation? Why would half of the mice, genetically identical, clonal, came from the same Adam and Eve in the freezer, and yet they behave differently? And so one of the things that can differ, of course, is during gestation where their position is within the uterus. So there's the uterine horns, and they can be at different locations. And so they can also be affected by the sex hormones of their surrounding pups. So are they females surrounded by two males? So there's a lot of variation that can occur during development that could contribute to this. But one of the things that I think is an obvious source would be the microbiome and how the microbes, which can sense an environment, can then work on not just the genome, but the epigenome. That's the idea of the microbiome. So the epi means above the genome. So a genome is a heritable thing in the nucleus inside of the cell. But the epigenome is not as heritable and not as solid. It's more plastic. So there's lots of readers and writers of the epigenome, things that turn genes on and off. And those can all be affected by the environment. So the environment really isn't going to go in and mutate your genes unless you've got some like very strong radioactive compounds or X-rays or things like that. But the environment can go in and affect the epigenome. And I think that the bridge between the genome, epigenome is the microbiome, because the microbiome is producing lots of compounds in its repertoire of things that it needs to live. It produces serotonin, which is our neurotransmitter that we need. It's produced in the gut. That's where 50% of your serotonin is found. So it's amazing that we've had this coevolution of the human microbiome and the human organism.
I didn't realize when you have a litter of mice that the sex of the pups in there can affect the other ones. Like, that is truly wild. Okay, so talk me through how you try to determine how the microbiome is impacting in addiction. What does that look like in your lab? And how are you trying to, I guess, nail it down so you can say as unequivocally as possible that you really think that the microbiome is playing such a huge role in this?
Yeah, one of the goals of our research program, this was specifically looking at cocaine use disorder. What we see is that there's a variety of trajectories that a mouse can take to becoming substance use dependent. And in this varying genetic population, there can be a variety of trajectories. And so we can sequence the genome of each of these diverse mice. They're called diversity outbred because 8 different strains were mixed together in such a way that each strain is now represented in each of the mice. Their 12.5% of the genome comes from one of the 8 parents in the crosses. Again, this is something you can do because we can control the mating and the environment. And so we have this population of genetically diverse mice, and we can take a little snip of tissue and using an array determine the genome and the genomic sequence of this particular mouse. And then we'll also be able to relate that to the behavior that we observe. And so now I just layer onto that simple program a layer of what does their microbiome look like. I'm mostly interested in the microbiome inside of the cecum, which is a part of the gut area that I'm interested in because I can actually look at the gut tissue associated with it. And there's a lot of issues with the gut-associated lymphoid tissue having immune factors that's going to be able to help with the exclusion or inclusion of different microbes in the microenvironment of the gut. And so I'm usually in the cecal contents and seeing what microbes are present there. And so I kind of layer that on to this gigantic correlation network of bacteria, behavior, genotype, or the genes at the different regions, and try to triangulate or try to find a model that best explains the data that we observe. And can we say that the data that we observe, for example, for the behavior, the use of the cocaine, quickly using it, very rapidly acquiring the trait of use and not wanting to get rid of that trait, not extinguishing very well, can we see if that trait is mediated by something in the microbiome? So is it an interaction between the genes and the microbes together? And if it's mediated by that, that would help us to understand the network that controls this behavior. Does it involve a microbe or does it not? In the beginning, I was looking at just kind of tagging the microbes using this process called 16S. It's a ribosomal sequence, kind of like a barcode for each of the microbes, and we'd figure out their barcode, and then you could count them. Then we could look them up in a tree and see how they're related to which types of lactobacillus, et cetera. We could name them. But basically, they had a barcode, and I could associate now the barcode with the different behaviors. But it turns out that that's not the most useful because the human microbiome is not going to be composed of the same microbes as the mouse microbiome. Humans have our own set of commensals that live with us, but they're going to be conserved at the level of function. So the bugs that I find with that barcode in mouse are doing a certain job, producing certain metabolites, and there's going to be a parallel or another bug in humans that's doing the same job. So we have to translate at the level of bacterial function, not just this barcode ID. And so that's where we can get into guilds of microbes working together, and then the metabolic composition or the hologenome, the genome of the microbes plus the genome of the host together, and how that's affecting a behavior.
How much do you have to think about how other microbes affect the microbes you're looking at? Right? You were saying you look at one particular part of the gut. Do you have to take into account microbes that are nearby that do different things?
Yes, you should, but in my reductionist approach, trying to find the simplest situation, eventually, with infinite money and time, we could expand to look beyond. But right now, we're picking one area where we can have some tissue from the host and then some microbes together and looking in that area and seeing the relationship between the communities there. So are there guilds or groups of microbes that are working together to do this function? And then what are the same ones that are in humans? One of the things, again, that's great at the Jackson Lab is because we produce mice to send around the world, we have very high biosecurity in that we monitor what is in the microbiomes for pathogens all the time. So there's no hantavirus running loose in the Jackson Laboratory. There's no other pathogens because we sell our mice commercially. So we offer these specific pathogen-free microbes. So they kind of have a defined or a limited flora as to which microbes will not be present. So we look by exclusion for microbes and we keep them out of there. So by having this regulated hierarchy of cleanliness at the Jackson Laboratory, some labs you have to gown in and out of and an air shower to get into in order to work that day, just because they don't want to bring any street bugs out, on your clothes into the room and inoculate the mice. A lot of mice have severe combined immunodeficiencies, and so they are very susceptible to things that are even not pathogenic. But working at the Jackson Laboratory, I'm able to leverage this specific pathogen-free facility and use it so that I can kind of control as much as I can the environment. And so we know which other bugs are not going to be present. We sterilize our food, we sterilize the bedding and their water and everything. And the light cycle is controlled. We control as many things as we can so that we can detect the microbial variation because it could get hidden behind things like diet. In the human population, when you're looking at the microbiome, it's so hard to make any comparisons or discover anything because everybody's eating different things and they've got different substrates to live on. Whereas here, I'm able to control for that particular aspect of the study.
So this strikes me as a perfect example of really basic science research that advances knowledge, but also has such huge potential for rethinking how humans interact with the world around them, including their microbiome that we don't really think about, which is really interesting. I'm curious. I feel like I jumped ahead more than I usually do, skipping all of your graduate and postgraduate work. Was this the type of thing that you found while you were in graduate school, or is this an extension? I mean, the microbiome is complicated. It's hard to just happen upon it and be like, I've got this great research idea. So I'm going to make you go backwards. Like, how did you get to this point?
Well, back at UMF during my senior year, and I did UMF in 3 years. Once I discovered that there was more college to have, I tried to get undergrad out of the way. And so I got the undergrad out of the way, but during my microbiology class, I somehow had picked up a pet iguana during my time in college, and I was interested in the hindgut fermentation of the iguana. So for a microbiology kind of independent study, I went ahead and I was culturing both the aerobic and anaerobic microbes in the feces of my pet iguana as a side project. And again, the independent study aspect of it was great because we didn't really have anybody doing research that I could join their lab. And there were some people doing some ecology research, and they've actually grown more and more with their molecular work that they're doing up there. However, at the time, I wasn't interested in looking at muscle diversity or lichens, but was very interested in, microbes and DNA. I was one of the first people to do a PCR reaction, which is the way of amplifying DNA using the thermocycler at the University of Maine at Farmington. They bought this piece of equipment. Nobody knew how to use it. And so I was working with Dr. Robinson, trying to learn how to do PCR, how to dilute primers. And it was very independent and self-motivated. You had to really, do it yourself. And so I like this freedom of being able to go investigate what microbes were growing in the gut of my iguana and then compare it to the literature. And historically, old naturalists have been studying the microbes of iguanas. And so I was looking at old papers, going to the library and getting interlibrary loan and things like this, and seeing how my results, what I'm seeing compared to what was observed. So nothing groundbreaking, but for me, it was a lot of fun. You can see some hints possibly of my downstream interest in feces in my undergraduate. But then when in graduate school, I actually was working in the most molecular lab I could find at Marquette University in Wisconsin, which was studying Arabidopsis, which is a plant. So I was looking at the simple genome of the plant. It only had 5 chromosomes. They were mapping that very easily. They named things like AT4G12470 was the name of a gene because they just named it based on identification throughout the genome. It was very logical time. And so I was very interested in, again, the impact of nature and nurture, the environment. How does the environment affect the genes? And so I was studying a process called vernalization. That's where plants need a winter in order to flower. And so we were studying a particular gene that was involved in kind of the hardening process. So not really the vernalization, the flowering process, but just getting used to and adjusting to the fact that it's cold. It's been like a cold snap. We haven't had a full winter. It's not time to go ahead and bolt and produce a flower yet. It is still just regular times, and we've just adapted. So environmental response, and I figured out some calcium signaling that was associated with this and the expression of different genes that went up just from temperature. So the temperature, cold treatment of plants, and genes were changing. So I found that extremely fascinating and tried to chase after the regions of the genome that were regulating that. What transcription factors were causing the genes to come on and on that were able to sense temperature.
What's it like to go from plants to animals? And do plants have the same issue with microbiomes?
Yes, there's definitely lots of interest in the plant microbiomes, especially associated with the agroeconomics of growing plants. One of the most interesting things is at the time, the best way to transform or to gene target plants was actually through the use of bacterium. So there's this bacterium called Agrobacterium tumefaciens, and it produces crown gall tumors on plants, but it's an example of transkingdom sex, where tDNA, transfer DNA, goes from the bacteria into the plant genome and incorporates in the genome. When I was making my knockout plants or things, I had to dip the plants in the flowers into a bacterial solution, and then the bacteria would follow the same track that a pollen tube would to inoculate the seeds. The bacteria would go down in and inoculate the ovaries of the plant and transform the DNA there. In undergrad, I was very interested in evolutionarily the whole endosymbiotic theory, the idea that our mitochondria and chloroplasts were once free living organisms. So they lived outside of us, and now they are dependent on us and we're dependent on them. So they used to be able to live without us, but now some of their genes that are needed to make a mitochondria powerhouse are actually in our regular human genome. So we are carrying some of their genes, so we're now codependent on each other. But I was very interested in the work of Lynn Margulis and in the endosymbiotic theory.
So you haven't found it difficult or confusing to go from plants to animals because for you it's just the gene. I don't mean just the gene, but like because you're looking at such that granular level, it's almost irrelevant.
Yeah, for me, it seemed like it's A's, T's, C's, and G's. They're doing different things in different organisms. And I mean, my plant can't move, but it still is able to respond to the environment. And then when I went to the Jackson lab, I came in as kind of a free agent. I could kind of choose which lab I wanted to work in. And a funny story I was telling my advisor, I've been there for several decades now. He's retired. And during my talk, he left during the middle of my talk. And I was like, well, that's rude. But then I found out why. He had to go home because his chicken coop was on fire. Seriously. And his wife called and there was an emergency to go take care of the chicken coop. And I was like, okay, I think I'm going to work in this guy's lab because he has an appreciation for like work-life balance. The fact that this thing was doing was important, but at the same time, he had obligations back home. And I wanted to have more of a well-rounded life and not be just dedicated to career alone. And so I was like, it shouldn't have been perhaps my deciding factor, but I joined him, and that's when I started to learn about immunology.
I think that's a fine deciding factor. So where are you at with the microbiome research now? I'm curious if you have a goal to be like, I want to know this about how the microbiome interacts in the next year.
Yeah, it's a good question. I have several little side projects, and I hope that one of those microbiome side projects gives me an answer towards this. Like, have I identified a gene that controls the abundance of a particular microbe? And I believe I have. And then I've also shown that that microbe, when I give it to mice, it'll affect their cocaine behavior. When I give them a compound, a metabolite that the microbe produces, it'll mimic giving them the microbe. So perhaps I've found the metabolite that's actually produced by the microbe that is going ahead and affecting their cocaine-related behavior. And so that's very interesting to me. And so trying to shore up those things and publish them in this coming year, make the findings clear and accessible to an audience, I think is an important next step.
I meant to ask you this earlier and I forgot. Why did you pick addiction to look at?
So yeah, it's kind of like addiction picked me. I didn't pick addiction. We had a new investigator come to the laboratory, Alyssa Chesler, who had a program at Oak Ridge National Labs, and they shut down their mouse facility there after decades of work. And so she brought with her a very extensive bioinformatic or in silico biology program and was looking for somebody to add some in vivo mouse testing to it. And she put out an ad and I answered it. I said, this is exactly the kind of thing I like, make hypotheses using data that we already have and then test them in the mice. And so that's kind of when I joined her lab. And she also brought with her a ton of microbiome data that she had collected at Oak Ridge National Laboratory as part of a Department of Energy program.
That's very cool. What we've learned about addiction in the last couple of decades is really extraordinary. And it seems to me, I think one of the reasons I jumped right into you talking about your research is because it's an approach and a thought process that seems even further beyond what we've started to really understand about addiction. And it's a really fascinating way to think about it.
One thing that I just wanted to interject is that in the United States, health in general can be predicted by zip code, right? The lifespan in southern Maine is, 5 years longer than the lifespan in, Washington County, Maine, just based on zip code. Now, is that because of the hospitals in the area? Is that because of the food deserts, perhaps? The fact that the southern Maine has a Whole Foods or a Trader Joe's and down east, we don't have one. We don't have the access to the Whole Foods. Is there an association between what you consume and your microbiome? Yes, we know diet affects the microbiome. Are there food deserts where people are living on 7-Eleven based food, which might include a banana if you're getting lucky at a gas station as far as real foods? And so I just feel like, making America healthy again perhaps could start with, looking at what food we have available to the population and how it's affecting the microbiome, which is then affecting our behaviors.
Yeah, that's a really good point. And the other part too is with the zip code, it seems like also the difference between doing manual labor and not manual labor, which really wears your body down as well.
Yeah, I agree. But from the point of the microbiome, a lot of the manual labor people are going to have a more diverse microbiome than somebody who's living in a pretty sterile office. I mean, we're antimicrobialing everything in our offices nowadays, from the soap and the wipes and everything. But if you actually work in the field, you are getting a lot of microbes. An interesting thing, a diabetes researcher who's retiring at the Jackson lab was saying, autoimmune diseases have gone up a lot, but what's happened at the same time is that our infections of pinworms have gone down. It used to be that pinworms were a childhood infection, kind of common in the United States, still common in many other places in the world, but they're not as common anymore. And so we've kind of gotten cleaner at home, but given our immune system maybe less to work on and perhaps become more autoimmune. And so that's the hygiene hypothesis that exists out there, suggesting that the cleaner we become, the more bored our immune system becomes and we start attacking ourselves. And that becomes personal for me because I have celiac disease where the immune system is interested in gluten from wheat that decides to attack my cells because of some antigen that's found inside of the wheat.
I'm mesmerized by your ability to think about all the different variables, but then also recognizing that you have to winnow it down and just figure it out piece by piece by piece. It's actually kind of a perfect example of how science often relies on small realizations and small aha moments, not like, oh, I've got it all figured out, but they just build on each other. It has been a ton of fun to talk to you. I could take up hours and hours more of your time, but I have a feeling you have to pay the bills and not just talk to me. Thanks for your work, and thanks for being so willing to jump into this conversation without me giving you any prep whatsoever.
Appreciate the opportunity. It's something I'm really excited about. If anybody ever sees me, just ask me questions. You can usually get me started talking and can't get me stopped.
Thanks for listening to the Maine Science Podcast. Make sure you never miss an episode by following us on your podcast platform of choice. And please leave a rating and review. It will help more people find us and help spread the word about some of the remarkable people doing science in Maine. The Maine Science Podcast is recorded at Discovery Studios at the Maine Discovery Museum in Bangor, Maine. The Maine Science Podcast is executive produced and hosted by me, Kate Dickerson, and edited and produced by Scott Loiselle. And the Discover Maine theme was composed and performed by Nick Parker.